Hello dear student, I am Dr. Ashok K.Yeole, Assistant Professor from Department of Zoology, KTHM College Nasik. In today's presentation, we are going to discuss in detail a topic of mammalian histology, and the topic is histological structure of ileum. So what are the learning objectives of today's presentation? First, what is ileum? Then what are histological structure of ileum and what are different functions of ileum. So let us start from what is ileum. Ileum is a posterior part of small intestine. In a previous presentation, we have seen a small intestine is divided into three important parts such as duodenum, jejunum and ileum. Duodenum is an anterior C-shaped structure of small intestine, then middle jejunum and posterior ileum. So small intestine is a largest part of alimentary canal, or we can say digestive tract. It is a tube-like structure in which the digestion as well as absorption takes place. Ileum is about 3 meters long and it starts from jejunum and ends to cecum. The small intestine is covered a part of duodenum, jejunum and ileum, mostly in higher vertebrates like mammals, reptiles, birds. The ileum is a part of small intestine is well developed. Even in case of fishes, this ileum is well developed because of the digestion and absorption takes place. The small intestine part ileum is a flat circular folding having a numerous lymphatic secondary follicular organ called as Peyer's patch. So Peyer's patch is a peculiarity of ileum. It is nothing but a follicular organ or a follicle of nodule, and the main function of Peyer patch is immunological in function. The inner wall of ileum consists of many folding-like structure, and that folding-like structure is called as villi. So villi is always bathed in a lumen of small intestine. So whenever food is passed from mouth to anus, it is temporarily deposited in a different compartment such as stomach, small intestine and large intestine. So whenever food is deposited in stomach, after semi-digestion, it is transported to a small intestine. So whenever food deposit in a small intestine for further digestion, it is always comes in contact with villi. So these villi are important. At the tip of villi, there are again many microvilli, and so these microvilli are secretory as well as digestive in function. It will help to release certain enzyme into the food as well as the important nutrients absorbed from the food is also takes place through these microvilli. Then histological structure of ileum. Generally, in a duodenum, we have seen histological structure of duodenum showing four distinct layers, that is mucosa, submucosa, muscularis and serosa. Same like duodenum, ileum also having a same basic structure, that is four distinct layers: mucosa, submucosa, muscularis and serosa. Only difference is that in case of duodenum, there is a releasing certain digestive bile juices and pancreatic juices in the small intestine, while in ileum no such a entry of digestive juices from different glands into the small intestine, but further digestion of fatty acids, or we can say lipids or fat substances, takes place in ileum. So first part of ileum is mucosa. So mucosa is innermost part of ileum. It bears, or the wall of mucosa bears foldings, and that folding is nothing but villi. Mucosal layer is highly lined with simple columnar epithelium. Mucosal layer lined with connective tissue also, and in between the lamina propria and lamina muscularis is incorporated by a mucosal layer. The characteristic feature of ileum is Peyer's patch. So what is Peyer's patch? Peyer's patch is nothing but an organized lymph nodule, and it is lined in a mucosal layer of ileum. Peyer's patch, which differentiate ileum from duodenum and jejunum. So in duodenum there is a C-shaped structure, and that C-shaped structure of small intestine leads into jejunum, and after the jejunum there is a starting 3-meter long tube, that is nothing but ileum. So these ileum bears a Peyer's patch. So these Peyer's patch are immunological in function. Then the intestinal villi is there. So these intestinal villi are nothing but finger-like projections, and these finger-like projections, that is intestinal villi, which are lined by epithelial tissue, and at the tip of these villi again bearing a many microvilli. So these microvilli secrete as well as absorptive in function. So in this slide showing a transverse section of ileum, as I said, transverse section of ileum showing 4 distinct layers, that is mucosa, submucosa, muscularis and serosa. So mucosa is the innermost layer, and it bears a finger-like structure that is called as villi. It is most important part of ileum. It covers maximum area. Then second part is submucosa. Submucosa, it is lined by a longitudinal and circular muscles. But in between a muscularis layer, these longitudinal and circular muscles are moreover concentrated, and the outermost layer is serosa, or it is also called as adventitia. So adventitia, muscularis, submucosa and mucosa. So these are the 4 important layers showing in the diagram clearly. Again, this diagram showing a Peyer's patch. As I said, Peyer's patch is a peculiarity of ileum. So the area of muscularis showing a lymphatic nodule, and that lymphatic nodules are immunological in function, and it is the most important characteristics or peculiarity of ileum. Then second part of ileum is submucosa. Submucosa is highly condensed with blood vessels, lymph nodules, and Meissner's plexuses with a connective tissue. These submucosa help to absorb nutrients from the lumen of the intestine, because whatever food is digested in the form of fatty acids and glycerol, it is to be absorbed from the digested food into blood by these submucosa. Then third layer is muscularis. Muscularis is highly retracted organ of small intestine. It consists of circular and longitudinal muscles. At inner side there is a circular muscle while outer side longitudinal muscle. So these opposite rotation of circular and longitudinal muscle help to create a pressure on small intestine. So these pressure exertion on small intestine, there is a peristalsis movement occurs. These peristalsis movement help to digest food into the small intestine. That means it provide a mechanical pressure on the food, so food become digest. So in small intestine, mechanical as well as chemical digestion takes place. So this peristaltic movement, or we can say mechanical digestion, is takes place in the small intestine with the help of this special area, that is muscularis, because this area is highly lined with circular and longitudinal epithelial muscle. Then fourth layer is serosa. It is a outermost layer. It is lined by simple squamous epithelium and connective tissue. This thin layer of ileum provide elasticity to the ileum. That is why whenever food is digested into the small intestine, it is to be moreover moving from small intestine to large intestine. So at this time, these serosa are playing an important role again. Then what are the different functions of small intestine? As I said, small intestine part, that is ileum, performing dwelling function, that is digestive as well as absorptive in function. So let us discuss first one, that is absorption of nutrients, vitamin B12, etc. Generally, what we observe in a stomach, complete digestion of protein as well as carbohydrate is takes place. But fatty substances, or we can say lipid, it is not digested into the stomach. So the digestion of fatty substances or lipids takes place in the small intestine, especially it is takes place in a part of lumen. So whenever the digestion of lipids is takes place into the lumen of small intestine, that is in lumen, it is in ileum. So it convert into fatty acid and glycerol. Fat is nothing but a complex substance, and it convert into simple form, that is fatty acid and glycerol. So these fatty acids and glycerol are the nutrients which present in the lumen of ileum, and it is to be absorbed into the blood via blood vessels which are present in submucosa. Along with fatty acid and glycerol, vitamin B12 is one of the most important nutrients which are present in lumen cavity of food. So it is again absorbed by the small intestine part, that is ileum. Then second function, as I said, Peyer's patch is important characteristics of ileum. It is nothing but immunological in function. So whenever the Peyer's patch is located in a submucosa area of ileum, it pick up antigen from intestinal lumen and transport them to the antigen presenting cells of a blood. So whenever antigen presenting cells receive these antigen, it shows a immunological function, because the another function of small intestine, that is ileum, is to prevent the growth of pathogenic bacteria in the intestine. So these three important functions of small intestine, that is ileum. So first one, absorption of nutrients, like fatty acids and glycerol along with vitamin B12. Second function, that is Pairs patch function. Pairs patch are immunological in function. It pick up antigen from intestinal lumen and transport it into the antigen presenting cells. And third function, it provide a immunological function, or it provide immunity, that is to prevent the growth of pathogenic bacteria in the intestine. So in this way you have to write in a short in exam. Generally in exam question arises, write short note on TS of ileum, or write short note on structure and histological structure and function of ileum. So you have to draw a diagram of TS of ileum, then enlist the structure, means you have to explain four layers of in ileum, and briefly mention the three functions of small intestine, that is absorption of nutrients, uh, fatty acid, glycerol along with vitamin B12, immunological function of Pairs patch, and to prevent the growth of pathogenic bacteria in the ileum. So this way we have finished this topic, that is histological structure and function of ileum. Thank you for watching this presentation. Next time we again meet with new presentation. Thank you.